for a month.
You've been living here for a month in our kitchen.
Yes.
So you're a scientist?
That's what they tell me.
Yeah.
Yes,
What does
I
that
am
mean?
a scientist. Officially, I'm an evolutionary biologist, and I do experiments. So even worse, I'm sort of this slightly crazy breed of evolutionary biologists where I actually watch evolution happen in the lab. And I use microbes so that I can watch that happen in real time, because microbes evolve quickly, because they reproduce quickly. What does being a scientist mean is sort of a different, bigger question. I guess I think of being a scientist, or how I think of being a scientist, versus the job description that the university probably has in my contract, is I think of it as telling stories. So you're trying to figure out the way the world works, and what you do is you propose a story, and then you test it, and then you're wrong. And then you propose a modified story, and then you test it. And because what I'm interested in is how evolution works, or how natural selection works, what I do is I try to make these narratives about the rules that natural selection is playing by, and how those rules play out in the real world. So it's a bit like writing a choose your own adventure. You're just trying to see how it all fits together, and that's what experiments are. You need this capacity to look at the world and then propose an explanation other than the ones, or in addition to the ones you already know. So you need to imagine something that doesn't yet exist, and then figure out a way to ask the world if this is, first of all, possible. Could this exist? That's like a lab experiment, or computer simulation, or mathematical models. You're asking, so I've got this crazy idea. Could this exist? Is this possible? And if you're very lucky and the answer to that is, why, yes, it could. Then you go out into the world and you ask, does this exist? Which is a different question, because not everything that can happen does happen.
Okay, so what I'm hearing is that there are some questions that are appropriate to ask as a scientist.
Yes.
And others that are not. How do you identify those questions that are appropriate to ask from a scientific point of view?
So science, at least experimental science, the kind of science that I'm familiar with, you can really only ask a question that you can falsify. So you can really only ask something that you can test, and where you can tell if the answer is yes or no. And most importantly, you have to be able to tell if the answer is no. So you're proposing a way that reality works, and then you have to find a way for reality to answer back at you. So a question that you can't test isn't a good question for science to ask.
For example?
In the short term, it doesn't mean science can't figure out. Sometimes you spend years just figuring out how to ask a question, right? So it doesn't necessarily mean the basis for that question is not answerable. You just maybe haven't figured out how to ask it yet, right? So when the question my lab asks, the sort of big question we ask is, can we predict what evolution is going to do? Can we predict it beforehand? Not can we do an evolution experiment and then explain it, but can we, without doing the experiment, predict what's going to happen? And then of course we do the experiment and see if our prediction is right. So that's like the big question my lab works on is evolution predictable? Then you get these little questions like, well, what would I need to know to predict it? What level of uncertainty am I happy with in a prediction? What about evolution am I trying to predict? You can't predict all the things you can't predict everything in perfect detail all the time. So you're saying, well, what bits of evolution do I want to predict, and with what certainty? How often am I willing for this prediction to be right versus wrong? Which bits are inherently unpredictable? So these are all questions you can ask, right? But you can't ask about a bit. You have to say, can I predict the direction of change? So can I predict whether a cell is going to get bigger or smaller? And so you really narrow down the questions into these incredibly specific case studies, and then you blow them up again and try to generalize from the case studies. Does that make sense?
I get the bit about narrowing down to set up an experiment with certain conditions to test whether something under those conditions is going to get bigger or smaller. But when you say you blow it up again, what do you mean?
So part of that is, so let's say I do an experiment, as I often do, where I take microalgae, these single-celled plants that live in all aquatic environments, And I evolved them under conditions of high CO2 or high temperature. So some conditions that are associated with global change. I'm not trying to mimic global change exactly. That would be impossible. I can't put the ocean in a test tube. But what I can do is I can say, okay, in response to these parts of global change, what do I see? Do I see cells getting bigger? Do I see them photosynthesizing more? Do I see them, for example, changing their strategies? Do they have sex more or less often? Do they clump together? So you start off sort of by doing natural history, which is you just start off by looking, saying, and then you come up with a question. So you haven't done the experiment yet. You've sort of done what we call pilot studies. And this is where you look. You can't answer anything because you haven't asked anything, the pilot study. You're just trying to figure out what to ask. Then you ask your question. You say, okay, if I put these under high CO2, I get transiently with the cells I look at, which is something called Ostriococcus or Chlamydomonas. Those are the two things we've worked with. You put them in high CO2, they eat carbon dioxide. So for them, this is like a buffet of goodness, right? And they get big and they grow fast. And the question I've been asking is, okay, if they get big and grow fast right away, so this is not evolution, does that mean they're going to evolve to be big and fast? And the answer turned out to be no. Actually, they slow back down. And so evolution spends all of its time reversing this initial response. And you're like, okay, so my first hypothesis or my question was, can I use this short-term response to predict evolution? And as so often is the case, evolution said, no, you cannot. Go away. Stop trying to ask that. And you're like, okay. But what we discovered is what we could predict. If we knew how much they responded initially, we could predict how much they would evolve. And the generality of that would be sort of saying, okay, we can't predict what is going to evolve. But if you think about how organisms are now as being the center of a circle, here, and how they're going to be in the future being a distance from that, what I can tell you is how big the circle is. Now that's really important because it means when we look at organisms now, a lot of the things we rely on them for means that we rely on their function being consistent or at least predictable. And what we found out so far is I can tell you how close to how they are now are they likely to be in the future. What I can't tell you is how they're going to be different, right? And so it seems like in some ways it's very disappointing, but in other ways we've learned a great deal. We've learned from these short-term responses, you can get a distance but not a direction. And that's actually better than nothing.
So how do you measure the distance in time, or?
Yeah, so distance in time is how much evolving they do.
But you don't know how they're evolving, whether they're getting bigger, smaller, thicker-skinned or thin-skinned.
Well, you can measure how they're evolving, but we can't predict it. So we can measure it. We can do the experiment and measure it and explain it after we've seen it.
Yes.
But this doesn't help us if we want to predict how things are going to be in the future, because what that says is what we have to do is wait for the future to arrive and explain it.
But you can predict that after a certain time there's going to be this distance. I'm just trying to get my head around the idea of distance.
Yeah, so it's sort of like saying, I can't tell you whether, it's like if you put a plant on the table, you say go away for a month, when you come back the plant will be 20% different. So that means it might be 20% bigger, it might be 20% smaller. I can't tell you if your plant is going to get bigger or smaller.
Okay.
But I can tell you when you come back it will not be the same.
I see.
So
I immediately think sort of difference in terms of there's been a whole lot of reproduction and replication of in some sort of direction you don't know what. So are you measuring it in terms of generations of?
So you're measuring change in fitness per generation, and fitness is
they've survived.
Yes, so
therefore they're on your radar and you can measure them.
So fitness is this terrible term. It's the basis for evolutionary biology. We know that natural selection acts on fitness, and fitness is really easy to define and almost impossible to measure. And it's just the number of surviving offspring that
it's the same
that is left, right? So you're like, yes,
you either die or you don't.
But when you want to measure it, you say, well, so are we looking at reproduction? Are we looking at survival? Are we looking at surviving reproduced, and you often can't measure survival and reproduction directly. So you're like, well, what do we measure? Population growth rate for microbes? You're like, okay, well, we have small population, now we have a big population in the same amount of time. But we're doing this in the lab, so in the real world, their survival will also depend on how well they can compete with other organisms, whether or not they get eaten, whether or not they get lysed by viruses. So all of these things will contribute to fitness, but in the lab, you just measure how long it takes them to divide,
Yeah.
right? So you come up with an experiment. This is the fun of doing stuff in the lab. You say, okay, in the real world, it's really hard for me to figure out what I should measure to measure fitness, because maybe you can't measure viral lysis in the ocean, or maybe you can't measure grazing in the ocean, in these microbes. But in the lab, you can set up an experiment where all you're asking them to do is grow fast. You're like, I'm going to change the environment. I'm going to give you lots of CO2 or a little CO2 or low pH or high temperature, and all I'm going to do, the only thing that matters your survival is how fast you can reproduce. And there you have it, you've defined fitness. And you're like, fitness is the rate of reproduction. Excellent. And you sort of simplify it in that way. And of course that means when you generalize, you have to start thinking about, okay, in the wild, maximizing fitness might not mean maximizing this rate of reproduction. It might mean avoiding being eaten. Right?
Yeah.
And so maximizing fitness may take on a different mask, a different actual phenotype. And so I think what I'm doing is trying to figure out trying to figure out the rules that natural selection plays by.